Another video tutorial from microbiology with Shaknik. So in that video tutorial we are going to discuss about a concept that is enzymes in RDT. Okay, so this video will literally cover all the important topics that you need to know from enzymes in RDT chapter. Okay, and remember one thing, that this is the basic. You need to learn about this very carefully. If you have learnt it very carefully, then you can answer any question from PCR. Okay, because they can ask you how you can label a probe in case of PCR. Okay, for that you need to know the basic knowledge of enzymes in RDT. Okay, and there are varying question how you can generate a primer in which the sequence is unknown to you. Okay, so in those cases you need to know about enzymes in RDT very well. Then you can choose appropriate tool for producing or labeling those unknown DNA sequence. Okay, so this is literally the basis of RDT, the first chapter regarding recombinant DNA technology. So I have already uploaded several videos regarding enzymes in RDT, where I have discussed about every enzyme in separate videos. So if you want to know about enzymes in RDT very well, so I would recommend you to go back into my YouTube channel and search for those videos, so that you can have a great knowledge for enzymes in RDT. Okay, although I am going to discuss about more or less everything here. So let's start. So we will start with the term recombinant DNA technology. Okay, so if we focus the term recombinant DNA technology, this recombinant DNA technology means here we are performing recombination of what? Recombinant DNA, right? That means recombination of the DNA, and here we are taking the help of the technology. That is the meaning of the science recombinant DNA technology. That means we are performing here the recombination of the DNA with the help of some technology. Okay, or for the development of technology, right? So what do you mean by the recombination or combination term? So we have various kind of DNA molecules, right? We have double-stranded DNA, we have single-stranded DNA, we have double-stranded circular DNA, we have double-stranded linear DNA, isn't it? And in such cases, with the help of recombinant DNA technology method, we can make a combination of them. Okay, in what manner? Let's come into that picture. Suppose this is suppose this is combination 1 and this is combination 2. Okay, so let's consider for now this is two DNA molecule, DNA number 1 and DNA number 2, and you are undergoing recombination and you have a mixture of DNA 1 and DNA 2. Okay, that is the basis of recombination DNA technology science. Okay, here we are just combining DNAs. For example, the most basic thing that we are doing with the help of recombinant DNA technology is, suppose this is our gene of interest, that is a double-stranded DNA molecule. Okay, and this is a circular DNA molecule. For example, let's talk about plasmid DNA molecule. Okay, so with the help of recombinant DNA technology, we can join them, okay, in such a way. And now suppose, let's think about that, you have a block named GI and you have a circle, okay, named pDNA. Okay, and now you have to add them together. Suppose you have a block made of paper and circle made of paper. Okay, and you have to join them together. So how can you join them? For joining you need some for joining you need some glue at that time, right? You also need some scissors so that you can cut those GI named paper from the paper section and pDNA from the paper section, right? And then with the help of glue you will add them just like that, right? So this is the thing that you have to perform when someone told you to make or to join your GI containing paper and pDNA named paper together, right? But in biology you can do this with the help of some tools which is known as biological tools, because here we are not you cannot use some scissors or some glues, right? So the scissors and glues for biology here is enzymes mainly, okay? These tools are known as biological tools, and these biological tools are nothing but enzyme that helps you to join a double-stranded DNA molecule named gene of interest with the plasmid DNA molecule, leads to the formation of that one. So this video lecture is totally based on what kind of tools or what kind of biological tools we need to perform all kind of techniques in recombinant DNA technology, okay? So I hope you have learnt well about the basics. So recombinant DNA technology is nothing but recombination of DNA molecule with the help of technology or for the development of technology, okay? And here with the help of recombinant DNA technology we have joined gene of interest and plasmid DNA here, with the help of which? With the help of enzymes, right? So now let's start our discussion regarding enzymes. So first of all I am going to discuss it with the help of some tables only, then we will enter about the details, okay? So let's start. First of all, enzymes in RDT. So first enzyme that we need to know is polymerase, okay? So although DNA polymerase, RNA polymerase and reverse transcriptase, I hope you know about all of them very well. Then another enzyme group is nucleus. So there are two kind of nucleus, endonucleus and exonucleus. Endonucleus can be further divided into two parts. One is restricted endonucleus and unrestricted endonucleus, okay? That means in restricted endonucleus the cutting will be restricted, but in unrestricted endonucleus there will be no restricted cutting. They can cut at any position, right? And exonucleus can be divided into two parts. One is when the DNA is degraded from the 5' to 3' direction, and when the DNA is degrading from 3' to 5' direction. Remember one thing, in endonucleus we are cutting the DNA molecule from the middle of the DNA, suppose in that one, and in exonucleus we are cutting DNA from any of the terminus, okay? That is the difference between the endonucleus and exonucleus. The third important thing that we need to know about the ligase, okay? That what I am telling about in that portion, okay, when I am trying to explain you the concept on the basis of a paper, that is the ligase. So this ligase is nothing but, remember, a biological tool that act like a glue, okay? How we are going to see. So this ligase will help us to join this gene of interest with that plasmid DNA in such a manner, okay? So ligase can be of two types. One is E. coli DNA ligase, another one is T4 DNA ligase, okay? So E. coli DNA ligase means the ligase which is purified from E. coli, and T4 DNA ligase means the ligase which is purified from T4 fudge. No, it is the ligase which is purified from E. coli infected T4 infected E. coli, okay? That means from the E. coli which is infected with a T4 fudge, okay? So that is the ligase, and we know that sometime when we are dealing with some too much supercoiled genomes, such as for human genomic DNA library, we need to recoil them, and we need to first of all uncoil them and recoil them. For those purpose we need some topoisomerase too. Okay, so here, and the most important part that we need is some modifying enzyme. For example, polynucleotide kinase that add 5' phosphate group with the help of ADP. What? Suppose this is a DNA molecule and you have added polynucleotide kinase PK, I am denoting, okay, in presence of ATP. So this PK will snatch the phosphate from ATP and convert the ATP into ADP and join that phosphate at the 5' position in such a manner. Okay, that is the idea of polynucleotide kinase, although we are going to discuss about this in a detailed manner within some minute. So then the second enzyme is alkaline phosphatase. Okay, remember the reverse work of polynucleotide kinase is performed by alkaline phosphatase. If polynucleotide kinase helps you to add a phosphate group at the 5' position of the DNA, then alkaline phosphatase helps you to remove or degrade that 5' phosphate group. Okay, so that is the entire funda. Suppose this is a phosphate containing group and this is a OH containing group at the 5' end. Okay, although we have OH at the 3' end in both the cases, but here we are discussing about 5' end. Okay, so if we want to come from 5' phosphate containing DNA molecule to 5' OH containing DNA molecule, you have to add what? You have to add alkaline phosphatase. Okay, that will just cut the phosphate, as its name suggests, the enzyme that cleaves phosphate. Okay, and if you want to come from OH containing group into phosphate containing group, then you have to add your PK, that is polynucleotide kinase. Here I have denoted with the help of PNK, the meaning is same, polynucleotide kinase. Okay, so that is why I have written here that polynucleotide kinase and alkaline phosphatase is a reciprocal functioning enzyme. And another modifying enzyme that you need to know, that is TDT, and I would personally recommend you to learn this very well, because this is a very important modifying enzyme that you need to remember. Okay, they can ask you how you can label 3' OH group, or how you can label your DNA molecule at the 3' end. Okay, and similarly you need to know about polynucleotide kinase very well. If they ask you that how you can label DNA molecule at the 5' end, you need to tell about polynucleotide kinase. At that time you will just tell about, or you will just mention that I will use polynucleotide kinase in presence of radioactively labeled ATP. Okay, so that the radioactively labeled ATP means it contains the P32. Okay, so the P32 will be simply snatched by polynucleotide kinase, converting them into ADP, which is no longer radioactive now, and attach them to the 5' end. That is how I can label 5' end of the DNA with the help of polynucleotide kinase. And if we want to label 3' end, then we should go for TDT. Now let's see why this TDT can label the 3' end. Okay, so although I have here drawn the picture, so this is, so you have to add terminal deoxynucleotide kinase in presence of only one kind of nucleotide. Okay, remember this very carefully. If you are using TDT, you have to add only one kind of nucleotide. It can be DGTP, it can be DCTP, it can be DATP, it can be DTTP, but 2 nucleotide at a time is not possible. Okay, suppose DGTP along with DATP is not possible. Okay, so the terminal deoxynucleotide transferase will simply add G nucleotides, that means the DGTPs, at the 3' end template independently. That you need to know very carefully. Okay, so that is how the 3' end labeling is going on, and we call them as homopolymer tailing. Okay, so remember another thing very carefully, that is the terminal deoxynucleotide transferase is an enzyme which is a example of template independent polymerase. Okay, you need to remember this about very carefully. So that is the outline of all the enzymes that we are going to discuss in the next 7 page. So let's discuss them about one by one. Now let's start our discussion with the template dependent DNA polymerase. So there are 3 kind of template dependent DNA polymerase, that is tag DNA polymerase, reverse transcriptase, DNA polymerase 1. Okay, so can we add here the TDT? No, because the TDT is polymerizing template independently, so we cannot add here, right? So DNA polymerase 1, also known as Conberg enzyme, it have, I know you know very well, better than me, about DNA polymerase 1, that is 3' 5' exonuclease activity, 5' 3' exonuclease activity, and 5' to 3' polymerase activity. But still, let's recapitulate. Reverse transcriptase, developed by Howard Temin and David Baltimore, RNA directed DNA polymerase that have the ability to synthesize DNA molecule from mRNA. Okay, very important for cDNA library preparation, RT-PCR, and for any kind of diagnosis where RNA is of interest. Okay, now let's discuss about that one, that is tag DNA polymerase. Remember, it is a very remarkable discovered DNA polymerase. Okay, because remember, without that tag DNA polymerase there will be no existence of polymerase chain reaction, PCR. I will going to upload a detailed video regarding PCR where we are going to discuss about everything at a glance. So without tag DNA polymerase there will be no existence of PCR, because remember, in PCR we are using, I have already uploaded video regarding PCR, I hope you have watched it. So in PCR we have 3 steps, right, in each cycle. So those 3 steps are denaturation, which is going on near about 90 to 93 degrees centigrade, then primer annealing, that is in between 50 to 70 degrees centigrade, and then the DNA polymerization, that is near about 70 to 75 degrees centigrade. So as you can see, there is a too much amount of temperature fluctuation within a fraction of minute, right? Because after 1 minute of denaturation we will go for primer annealing, which is of 1 or 2 minutes, then again the DNA polymerization step, which is also for some minute, right? So there is a too much fluctuation, and if we use normal DNA polymerase such as DNA Pol 1 or Pol 3 or reverse transcriptase, which is a normal reverse transcriptase, so they cannot withstand with those kind of too much temperature fluctuation, right? That is why the DNA synthesis in vitro is not possible. But tag DNA polymerase, which is from the Thermus aquaticus enzyme, which is a bacteria who lives in geothermal vent where temperature is near about 80 to 90 degrees centigrade. At that temperature, those tag DNA polymerase can stable and operate at 72 degrees centigrade, but that means it is optimum temperature, right? That means what? That means tag DNA polymerase can survive at 72 degrees centigrade and 92 degrees centigrade. That means what? We can now, after the discovery of tag DNA polymerase, we have now the independence to synthesize DNA in vitro. Otherwise, we need to wait for 3 to 5 to 7 days for in vivo DNA cloning, right? So that is the importance of tag DNA polymerase. I hope you can understand. So it has 5' to 3' polymerization activity and 5' to 3' exonuclease activity. Okay. So now let's come to the topic, that is nucleus. Nucleus means what? Nucleus means enzyme that have the ability to cleave nucleotide, right? As its name suggests. So mung bean nucleus, the first discussion, that is derived from mung bean sprout. Okay, so it is a endonucleus. Endonucleus means it will cut the DNA or RNA from the middle. So it cuts mainly single stranded DNA and RNA molecule. Okay, and requires Zn2 plus ion for their catalytic activity. So mung bean nucleus, it is a endonucleus and cuts single stranded DNA and RNA molecule and requires Zn2 plus ion for their catalytic activity, and it is purified from mung bean sprout. So now the next one is S1 nucleus. It is again a endonucleus and it is purified from Aspergillus oryzae, and it, as like as mung bean nucleus, also cuts single stranded DNA and RNA molecule. Okay, and along with that it can cleave a strand which have a nick at its complementary strand. Okay, so S1 nucleus and mung bean nucleus is quite similar, but S1 nucleus have a function in addition, that is it have the ability to cleave the strand which have a nick on the opposite or on the complementary strand. Okay, so now let's move to the RNase A. RNase A is an endonuclease that digests single stranded RNA at the 3' end of the pyrimidine residue. Okay, so now let's move about RNase H. This is endonuclease and it digests the RNA strand of the RNA DNA hybrid. Okay, so does not digest single stranded or double stranded DNA. So this RNase H, you can find RNase H in HIV virus replication. Okay, I have already uploaded a video regarding HIV virus. You can watch this if you want. So in that time, after the in that time the reverse transcriptase enzyme have two activity. It show the polymerization activity first, that result in the formation of a, suppose the black color line is a RNA molecule, that is the single stranded positive sense RNA, negative sense RNA molecule that they possess, and this is your cDNA molecule, that is that generated by reverse transcriptase. Okay, now RNase H will come into the picture and it will degrade the negative sense RNA molecule just like that. Okay, so that there will be only now single stranded DNA molecule. So we are now using that RNase H also for our own purposes, and it is a endonuclease. Okay, so now let's move on end modification enzyme. Another important thing, so although we have discussed it in a short manner earlier, so the first thing, end modification enzyme that I want to discuss with you, that is terminal deoxynucleotide transferase. So I have already discussed with you about TDT in a very short manner at the starting of that video. So this terminal deoxynucleotide transferase will label at the 3' end, right, as I have told you. So how it can do this? It can do this if you add simply a one kind of nucleotide. Suppose look here, I have only used the DCTP. So what it means? It means it will add simply the cytosine residues at the 3' end, right, resulting in the formation of a 3' staggered end. Okay, so it is also another important function of TDT that you need to remember, that is TDT also have the ability to form the 3' end staggered or 3' staggered end. Okay, and the process is called homopolymer tailing, because remember the 3' end are usually called the tail, and here we are adding the homopolymer, right? Homopolymer means the polymer that have only one kind of nucleotide. That is what we are doing with the help of terminal deoxynucleotide transferase, right? Now the second thing is alkaline phosphatase. I have also discussed about alkaline phosphatase in my at the beginning of that class. So alkaline phosphatase removed the 5' phosphate group, right, as we have discussed. So it can be isolated from E. coli and calf intestinal source. So remember, the alkaline phosphatase in case of E. coli is stable, but it is less active than the calf intestinal source. Okay, so there is a advantage in E. coli alkaline phosphatase and also disadvantage that you need to remember, and it simply cuts the 5' phosphate groups, right, and make them OH group containing. Now the question is, what is the utility of that alkaline phosphatase? So remember, when we are ligating our gene of interest with the plasmid, okay, there will be a situation called self ligation, meaning what? As the plasmid is also opened, okay, so in presence of ligand they show a affinity of self ligation. That means instead of joining with the DNA of interest, it will prefer to join with itself, right? But if you cleave the 5' phosphate group of the plasmid, okay, so can it self ligate? Yes or no? No, because as it do not have phosphate group, so there will be no phosphodiester bond formation with the help of DNA ligase, right? Remember one thing, DNA ligase join nucleotides by simply forming a phosphodiester bond between them. Okay, so we are going to discuss about DNA ligation. So as a result, but as still our gene of interest contains the phosphodiester bond, so there will be only ligation between the gene of interest and the plasmid DNA, right? So that is why it helps in prevention of the self ligation of the plasmid vector. Okay, so now let's come to the T4 polynucleotide kinase. Okay, we have also discussed about this in our starting of the class. So T4 polynucleotide kinase is isolated from the E. coli infected T4 phage. Okay, E. coli infected with T4 phage. Okay, so as I have told you that used for in-labeling of DNA, or if I want to say this more specifically, it is the 5' in-labeling of DNA technique. Okay, so it can be performed by both forward reaction and exchange reaction. If you can see about the forward reaction, in forward reaction we are simply adding the phosphate by taking it from the ATP, right? But in exchange reaction there is a exchanging. Okay, yes, there will be a exchanging, that is, it will first take the phosphate group and give them to ATP, and then it will again take the phosphate group from the ATP and add this with the 5' end. Okay, that is the forward reaction, exchange reaction, right? That means there is a exchange of phosphate between ATP and 5' end of the DNA molecule, as you can see. So now let's come to the ligase topic. So remember, DNA ligase joins two DNA molecules, right? And we all know there is a phosphodiester backbone present in the double-stranded DNA molecule, and in between them there is bases occurs as a steps, right, in ladders, and these red marked portions are what? Phosphodiester bonds, right? So ligase simply join two DNA molecule by synthesizing a phosphodiester bond between two nucleotides, okay, at the end of the two different molecule or at the two end of a single stranded DNA molecule. Okay, so as you can see here, there is a DNA backbone present, right? So there is a DNA backbone and there is a sticky end. Okay, sticky end. Why I am going to discuss about sticky end and blunt end in restriction in the nucleus video. So what it do? So remember, there are two kind of ligase present. One is E. coli ligase or Econi DNA ligase, and it uses NAD plus as a cofactor. Okay, what will occur? This NAD plus will simply split and result in the formation of E. coli DNA ligase AMP complex formation. Okay, that is AMP complex formation. And now this E. coli DNA ligase AMP complex will go and bind with the nick generated. Remember, if in two DNA molecule there is a absence of phosphodiester bond but presence of base, as you can see that bases here are present. Oh, I have taken the white color, sorry. Bases are present but no phosphodiester bond, just like here. So in such conditions are called nick. Okay, and those nicks can be sealed with the help of DNA ligase, and here DNA ligase sealed the nick by formation of DNA ligase AMP complex, in which in case of E. coli DNA ligase helps NAD plus. Okay, NAD plus helps E. coli DNA ligase to leads to the formation of E. coli DNA ligase AMP complex, and this E. coli DNA ligase AMP complex will go and bind with the nick. Okay, and remember there will be obviously a exposed 5' phosphate group and 3' OH group. Okay, suppose let's consider about this part as the 5' in and this part is the 3' in. That means there will be a exposed 3' OH group, right? And similarly here will be also a exposed 5' phosphate group, right? And the ligase AMP complex that I have denoted here as LAMP will simply help them to form a covalent bond, right? And that is the phosphodiester formation that leads to the joining of them. Okay, I hope you have understood it very carefully. So now, DNA ligase join DNA molecule together by synthesizing a phosphodiester bond between nucleotide at the two end of the two different DNA molecule, or in some cases at the two end of a single molecule, right? You can see in that picture it joins nucleotides by forming a phosphodiester bond in between them, right? That is the idea. So DNA ligase joined DNA molecule by forming a phosphodiester bond between adjacent nucleotide. Okay, and it can be between two separate DNA molecule, or it can be between the two end of a single DNA molecule, or single stranded DNA molecule, or a single double stranded DNA molecule, what you can see. Okay, so there are two kind of DNA ligase that you need to remember: T4 DNA ligase and E. coli infected DNA ligase. E. coli DNA ligase. E. coli DNA ligase is isolated from E. coli, and T4 DNA ligase is from T4 infected E. coli. Okay, remember one thing, that T4 DNA ligase uses ATP as a cofactor, and here E. coli DNA ligase uses NAD plus as a cofactor. T4 DNA ligase helps in ligation of both blunt end and sticky end, and E. coli DNA ligase only joins sticky end. Okay, and remember sticky end joining is easier than blunt end. Okay, so here I have mentioned what I have discussed you diagrammatically, that is in each case the cofactor is split, that is in case of E. coli DNA ligase NAD plus, and in case of T4 DNA ligase ATP, and form a DNA ligase AMP complex. This complex bind the nick, which must have 5' phosphate and 3' OH exposed group, and make a covalent bond in the phosphodiester chain. Okay, so now let's move to the another topic, that is Lino fragment. Okay, so I am going to discuss about Lino fragment with the help of that diagram. So what is Lino fragment? Remember, Lino fragment is the larger part of DNA polymerase 1. Okay, so DNA polymerase 1 has three type of activity, as we have discussed: 5' to 3' exonuclease activity I have mentioning here as 5' to 3' E. Okay,
and 5 prime to 3 prime polymerization activity, and 3 prime to 5 prime exonuclease activity. Okay, so here remember, the 5 prime to 3 prime exonuclease activity simply involves in the nick translation, okay, during the DNA repair. And the 5 prime to 3 prime DNA dependent DNA polymerase activity, that is nothing but the, okay, I think now you can see this. Okay, that is the DNA dependent DNA polymerase activity. And there is also the 3 prime to 5 prime exonuclease activity that mediate the proofreading and primer removal. Okay, so by subtilin digestion we are removing simply that part of the DNA polymerase that is responsible for the 5 prime to 3 prime exonuclease activity. That means that helping us by doing during the nick translation of the DNA repair. Okay, so we are removing that part with the help of subtilisin digestion. So our Lino fragment will lack the 5 prime to 3 prime forward exonuclease activity that helps in nick translation of the DNA repair, but it have 5 prime to 3 prime DNA dependent DNA polymerase activity and 3 prime to 5 prime exonuclease activity that helps us in primer removal and proofreading. Okay, so let's discuss about what is the use of Lino fragment in recombinant DNA technology. So recombinant DNA technology, it can produce for you blunt-end from staggered end. Okay, we are going to discuss about this how. So just remove the 2 point for now. We are going to discuss it, and it also helps you to prepare radioactive DNA probe. Okay, and it synthesizes double-stranded DNA molecule from single-stranded template. Now come here. Suppose this is the gene, this is your gene of interest, okay, and this is your gene of interest. Okay, so but unfortunately you have a plasmid vector that possesses blunt end. Okay, that means suppose you are a research scholar, your gene of interest has 3 prime overhang and 5 prime overhang, and you have unfortunately you don't have unfortunately any enzyme that can generate a staggered end at the plasmid vector. Okay, so as a result you cannot join them, right, because blunt end do not join with another sticky end, right? Blunt end will only join with the blunt end, right? So how can you join them? This is going to be a very difficult task. In that position we are going to use the Lino fragment. So what Lino fragment help us? Let's talk about 3 prime overhang. That means we have this kind of gene of interest. Okay, so in 3 prime overhang, the exonuclease activity containing Lino fragment will simply bind over here, and it shows is 3 prime to 5 prime exonuclease activity. Okay, that will simply remove the 3 prime or the preceded 3 prime end. Okay, that means the 3 prime end which is in excess amount. Okay, as a result a good 5 prime to 3 prime blunt ended DNA fragment will produce. That I have mentioned here in as a first point, that is digesting away any protruding 3 prime overhang or the 3 prime in nucleotide which is in excess amount. Okay, with the help of 3 prime to 5 prime exonuclease activity. And now let's see about what it does in case of 5 prime overhang sealing. In that case it will activate the 5 prime to 3 prime polymerase activity. Okay, and simply help in filling the preceded 3 prime end. Okay, that is how it helps in case of 5 prime overhanging. So in case of 3 prime overhang, it will simply remove or digest the protruding 3' end, okay. As a result, a good blunt-ended DNA molecule will generate, and in case of 5' end, it will simply helps the preceded 3' end to complete its synthesis. As a result, a good double-stranded DNA molecule or a blunt-ended containing double-stranded DNA molecule will generate. Okay, that is the use of Lino Fragment. Okay, very important for examination purpose. Filling in residue 3' end of the DNA fragment to make overhang blunt end producing, and digesting away protruding 3' overhang 3' to 5' exonuclease activity. And also it helps you to prepare the DNA probe, radioactive DNA probe, and also help you to synthesize a double-stranded DNA molecule from single-stranded template. Okay, so that is the concept behind the Lino fragment. Okay, so that is all kind of enzyme that you need to know in enzymes in RDT chapter. But remember, here we are not discuss about the main hero of recombinant DNA technology, that is the restriction endonuclease. So we are going to discuss about restriction endonuclease in detail in some another video. So I hope this video will be helpful to you, and if it is, please hit the like button, share with your friends, and subscribe to my channel to get more videos like that. And thank you for listening to this class.